I welcome all of you to today's webinar. I am Dr. Mahua Dasgupta Kapoor. I'm the Director of Medical Affairs for HaystackAnalytics. For all of you who are new to HaystackAnalytics, I'll just take the next 15 seconds to walk you through our journey. So basically, we are a very young Indian company who are there out in this world with just one mission, and that is to empower diagnostics, not just nationally, but globally, to bring the most advanced technology so we can diagnose better. And the diagnostic stewardship that we can establish will gradually translate into very effective antimicrobial stewardship. And that is so important given the context of the patient, because all that we are trying in healthcare is better patient outcomes. The journey has been short but very interesting for us. We started off in 2019 and here we are in 2026 with now different verticals in the healthcare system that we are working with. Ranging from personalized genomics to genomics in infectious diseases. We have our flagship products in place. Today is not the day to talk in details about the same. Today, because what we intend to spend the next hour is to understand the bug to drug. So in the book, The Art of War by Sun Tzu, it is so rightly mentioned that the fairest thing to do in a situation, in a combat, is to know your enemy well. Now, while this is too hard a statement to be made for microbes, which outnumber us in the world, not only that, they have a lot of usefulness and contributions to our own being in this ecosystem. Yet, for many of the pathogens which wreak havoc in our life, it is important for us to know them well if we ever intend to win the combat and stay ahead of them. So that's what we are going to hear from none other than Dr. Ayesha Khan. And it is such an illustrious and rich career that she has so far had in such a short span, I would say. She's the co-chief division of clinical microbiology at the University of California, Irvine School of Medicine. She's also the director for research and development and the assistant professor for pathology and laboratory medicine. Her expertise and focus are into antibiotic resistance, and especially in case of high-risk bacterial pathogens. The insights into how these microorganisms evolve and make sure that they can defeat our endeavor to control them through the use of antibiotics. What makes it very interesting is because her work involves translational research. So it is not only about knowing, but also bringing it to a point where there is a clinical application towards better patient outcomes for it. And that is what is she going to share with us. Very important professional contributions and leadership that she has exemplified in the field of making guidelines which all of us in healthcare have fallen back upon, especially the clinical microbiologists in the group would identify with this. CLSI, which almost is the Bible when it comes to reporting antibiotics for a disease for us, something now that even clinicians have started depending so heavily on. And she has guided a lot of research and made sure that there is rich literature available for referencing. So I don't think I'll take much of her time because I let her utilize this period with us so we get enriched by what she has to share. So over to you, Dr. Ayesha. At the end of your session, we'll go into the Q&A. Till then, the floor is yours and the audience as well. So over to you. Welcome to this webinar.
Thank you so much, Dr. Mahua. Thank you so much for having me and for thinking of me and considering giving me the opportunity to be here. I think it's sometimes I'm more excited to be able to connect with my community back home. I'm based in the US, but I'm South Indian, and I think it's rarer for me to be able to talk to Indian microbiologists and clinicians and pharmacists. So I think I'm very grateful. So I'll I changed the title a little bit to get people to start thinking about what this means and where this is moving. Because I think at the beginning there's going to be, a lot of data and mechanisms, but just in the back of your mind, I want you to remember that this is all working towards something bigger in terms of what we're trying to change and gaps that we're trying to fill in terms of what's the paradigm right now in medicine. Right now, in the clinical microbiology laboratory, we detect resistance after it emerges already, right after it occurs. And I think a lot of connecting the dots between different types of methods and now using artificial intelligence and deep learning models, et cetera, I think the field is moving towards trying to stay one step ahead and try to maybe even predict the likelihood of resistance emerging before it happens so that we can actually do something proactive about it. So just keep that in the back of your mind. I'm a clinical microbiologist by day, so I spend the day focusing on patient care. And so I'll start a little bit with that context. Just some disclosures, none relevant are having any bearing on my talk today. So like Dr. Mahua mentioned, I think starting contextualizing the problem and microbial resistance is obviously a major public health crisis. I think how we tackle that is we've kind of stayed within the box for the most part. But I think something that I've had a lot of appreciation over in my journey of understanding microbes is seeing them as teachers and they're the simplest forms of single-celled life, the first forms of life on Earth that are still by far, I would say, the most successful life forms on Earth. I think that goes to show that maybe multicellularity isn't exactly a reflection of complexity. So this is just an older, if you remember, this was a popular YouTube video that went around, but it was a science paper that essentially took a big petri dish and tried to show that antibiotic resistance evolution happens very quickly. So in this petri dish, there's different segments that are have different concentrations or gradient of the antibiotic, the outside being the least, and then the first layer being the exact concentration that's enough to kill a susceptible bacterial strain, and then 10 times that concentration, 100 times that concentration, and then 1,000 times that concentration. And what they're going to do is basically add bacteria that are susceptible to this particular antibiotic on the outside and look at how growth evolves and changes over time. And I'll talk a little bit about what the term MIC means later, but essentially you do see that there's some level of inhibition. They're struggling to grow at the beginning in the first quadrant. But very quickly, you do see that there's certain cellular populations, subpopulations that are able to eventually grow up to 1,000 times the concentration that they were initially susceptible to. And this doesn't, this doesn't take very long. This is a 10-day time lapse. And I think we can appreciate that essentially this is what's happening within patients as well, and or the scope and potential of this happening within a patient is what leads to, for example, development of resistance on treatment, right? And I think with each patient, at least I've gained a lot of respect for a single-celled organism's capacity, infinite capacity to and not just as single cells, but as bacterial communities adapt and survive. So a lot of the research that I'll talk about today is trying to understand on a molecular level how they're doing this and then translating that to the bedside. I will start a little bit with like our existing paradigm in terms of where we are in the clinical microbiology laboratory. So, patient comes into the hospital, whether it's the ED or if they're admitted. But generally, if they're based on their symptoms, clinical presentation, history, risk factors, let's say they come in with signs of bacteremia, empiric treatment is usually started. I'm giving an example of blood cultures because it's easier, but if cultures are collected, then there's a certain amount of time from the first time point of when empiric treatment is started. So blood cultures come to the lab. Generally, they're incubated and let's say it grows overnight and flags positive. And this is if you have an automated blood culture instrument, some places don't. then there's a gram stain. A gram stain is still by far one of the most clinically useful tools that's been has clinical utility for hundreds of years because what it does is it helps narrow treatment. So if you started with something that tried to cover for all potential causes of a pathogen, different types of pathogens that might be causing an infection, a gram stain looking at under a microscope allows you to see, oh, okay, maybe I can take something away, or maybe I need to actually add something more. From that point, now there's a lot of technology that has emerged to try to give you a little bit more detail as fast as possible and as accurately as possible. So maybe in about 3 hours, some places have PCR panels that they're able to run based on what they see on the gram stain that give you a little bit more information. And some of these panels are pretty comprehensive, so they can give you a lot of information. But ultimately, to actually get organism to grow, right? A culture, a bacterial culture, in this case, we're talking about bacteriology. That's what's necessary to be able to identify, confirm identification, and then ultimately perform gold standard phenotypic antimicrobial susceptibility testing. And this whole process can take, it's very variable, but ultimately on average about 2 to 3 days. But that is still the paradigm, that is still the gold standard. So we're going to talk a little bit about antimicrobial susceptibility testing and its limitations. So if you are somewhat familiar with what clinical laboratories do, there's different methods. People are probably the most familiar with disk diffusion. It's by far the most globally used manual method to determine if a organism is susceptible to a particular antibiotic. But ultimately, there's a number that we're trying to derive from some of these methods, right? So an example is the term MIC, which basically means minimum inhibitory concentration, which means what's the lowest amount of antibiotic that is needed to actually kill this particular bacterial strain or inhibit the growth of this particular bacterial strain. That number on its own doesn't really mean much, right? It's just a number. So clinical breakpoints are essentially what allows us to take that number and make it mean something in patients. So depending on what those cutoffs are, clinical breakpoints vary by bug, by drug, by infection site sometimes. But ideally, in a perfect world, what they're supposed to do is if I get a number, hypothetically 2, then based on these clinical breakpoints, let's say the breakpoint is anything less than or equal to 4 is susceptible, then that should tell you that if you get 2, then there's a higher likelihood that if the patient is treated with this antibiotic, then it will likely work. And the opposite is true for resistant. If it's something that is resistant, then it probably won't work. And then there's newer categories like susceptible dose dependent, which now account for certain types of infections and organisms that need a lot more antibiotics, so a higher dose of antibiotic, in order for them to have a better chance, in order for that patient to have a better chance of actually clearing that infection. So a lot of what I'll talk about today is what happens when that breaks down, right? That's a perfect scenario, but what happens when the susceptibility testing that we're doing in the lab is not very good or does not accurately predict clinical outcomes in patients. So for example, whatever methods labs are using are calling a organism susceptible to an antibiotic, but ultimately the patient fails treatment. So there's discordance or lack of correlation between the testing that we're doing in a tube in the lab. I mean, that kind of speaks to why there might be discordance, right? We're not recreating the conditions of the host, et cetera. So what do we do in that scenario? So this is just a use case. A lot of the projects in my lab right now focus on these organisms because they're so problematic. But I think the concept of understanding mechanisms of resistance and applying that to both diagnostic and therapeutic development applies in general to microbiology. So I'll focus today on talking about metallo-beta-lactamase producing carbapenem-resistant Enterobacterellus. It's a mouthful, so I'll refer to it from here on out as MBLCRE. I will talk a little bit about what they are. So these are under the umbrella of carbapenem-resistant organisms, which are, one of the higher scales of what is very challenging and difficult to treat in a clinical setting. These are probably the worst. They cause severe infections that can be intrinsically resistant to a lot of antimicrobials and then very easily acquire resistance as well. And there's not a lot of treatment options right now available. Initially, even for the longest time, and I think India is a regionally an example, along with a lot of other regions like Southeast Asia, Eastern Europe, where metallo-beta-lactamases have been the most common carbaminobases circulating in those regions. But there's been a very recent shift in epidemiology. So recently, there's been, within the last 6-7 years, essentially, there's been a pretty dramatic increase in global and regional prevalence, specifically of New Delhi metallo-beta-lactamase producing CRE infections. And this is specifically a graph looking at the United States. So by far, globally, KPCs have been the most common carbaminobases, but recently in some hotspots in the United States, like New York, Arizona, NDM enzymes as of 2021, around then, have overtaken, surpassed KPCs, and become the most common carbaminobases in those regions. So it's a very fast and quickly disseminating resistance mechanism, and the landscape epidemiology-wise changes very quickly. Like I said, a lot of the beta-lactam, beta-lactamase inhibitor development, so a lot of the new drugs that have been developed within the last 10, 15 years, it's focused on serine carbaminobases, so not metallo-beta-lactamases, but the other types of carbaminobases. So historically, we haven't had a lot of options. There really isn't anything that directly targets the metallo-beta-lactamases themselves. So recently, there's been some developments. There's in the last, again, 5 years, there's 2 recently approved new agents that are now the preferred treatments recommended for the treatment of MBL-CRE infections. They work similarly, but they're a little different. Cefiderocol came first, and then Astrena-Mavibactam. So Astrena-Mavibactam really mechanistically is a derivative of ceftazidime-mavibactam plus Astrionum, which is a homebrew combination people use with the same logic of combining Astrena-Mavibactam. You don't need to get too lost in the details, but just to keep in mind that whenever there's a beta-lactam and a beta-lactamase inhibitor, they have to work together. So Astrionum is a very old beta-lactam, but it's kind of weird and a little different in that it actually is stable in the presence of metallo-beta-lactamases, which chew up every other beta-lactam. So metallo-beta-lactamases can hydrolyze every beta-lactam except Astrionum. But it's also very vulnerable to some of the, in comparison, very weak beta-lactamases like ESBLs in comparison to MBLs. So it's still vulnerable to many of the other mechanisms that these cells often co-carry. And that's where avibactam comes in. So avibactam, originally paired with ceftazidime, is really one of the first new age beta-lactamase inhibitors that was really good at inhibiting multiple serine beta-lactamases at the same time. So in effect, it acts like a shield. Its job is to shield Astrionum and allow it to function. Cefiderocol is a little bit different. It is a beta-lactam at the end of the day, but what makes it different is how it gets into the cell. So antibiotics usually get into a cell by passive diffusion. If there's more outside, less inside, right? Very basic passive diffusion. But cefiderocol is a little bit different in that it hijacks, it's Cidara-4, so it hijacks bacterial iron transporters. So they have iron transport systems that they normally use to find iron in their environment because they need it to survive. So it's able to use that as a Trojan horse and hijack those transporters to actively be pumped into the cell. So now there's a lot more antibiotic in the cell than there would normally be. And it's relatively stable to metallo-beta-lactamases as well. And I say relatively stable because there has been emerging mechanisms of resistance that are metallo-beta-lactamase related to cefiderocol. So I think I always start with thinking about patients. So in the last 2 years especially, but in general in the last 10 years, there's been increasing reports of patients failing either cefiderocol or ceftaz-avi-Astrionum or Acinetobacter now that that's available. Failing treatment due to the rapid development or emergence of resistance during therapy. And there's a few case reports. If you look up, a quick example is a recent case report from a group in Stanford where they described a fatal infection, E. coli infection caused by an NDM producing E. coli, where a patient essentially failed treatment back to back. So this was a 12-year-old. Again, a lot of times when it comes to MBL-CRE infections, these are very vulnerable patient populations. They have a lot in terms of underlying risk factors. This is a transplant patient and was admitted for a sepsis workup. A PICC line was placed. Initial cultures were blood cultures that grew an NDM producing E. coli. Oftentimes now in clinical labs, because they're able to detect the presence of metallo-beta-lactamases in different ways, whether that's a PCR or a lateral flow device that they can perform directly on colonies. But a lot of labs are not testing for the newer agents like Acinetobacter yet, at least. We're not yet. So empirically, if there's an NDM detected, you have two options, right? So in this case, they just started the patient on Acinetobacter plus ceftazidime-avibactam. The patient did improve in the following days, and blood cultures, repeat cultures were negative, but then by day 26, cultures were positive again for an NDM producing E. coli. And at this time, at least in the United States, and I think this is very similar to a lot of other areas where especially for newer agents, last resort, last line agents, if the clinical laboratory is not performing in-house testing, then public health laboratories or reference laboratories are performing that testing. But the turnaround time is pretty long, so you can't really use that information to actively guide management and treatment decisions. So at this point, by day 26, the results of the initial isolate came back. So MICs were already pretty elevated to Acinetobacter 16, but the cefiderocol MIC was pretty low, 0.38. So based on that, they switched. And then they also had the MICs of the subsequent isolate after the patient was treated with Acinetobacter, when now the MIC went up to 64. So the patient was switched to cefiderocol. Source control, the PICC line was removed, but then blood cultures were positive again. And then this time, the last isolate was, so now high-level resistance to both Acinetobacter and cefiderocol. And this patient ended up passing away, and the family chose to withdraw care. Ultimately, though, I think it is on us to make every single patient matter in some capacity. So whole genome sequencing was performed to understand the underlying basis of resistance. And I'm just going to show you just in some animated cartoons, like what the ultimate findings were. So that this was an NDM5 producing E. coli. There's different variants. NDM5 tends to be pretty notorious in terms of being difficult compared to even other NDM variants. This isolate also had another beta-lactamase called EC18. These are very similar to your routine AMP-C class C beta-lactamases, CMY. They're not as thought about until more recently because they're becoming problematic. But based on whole genome sequencing, what they were able to show was the baseline isolate. So the first isolate before the patient was actually exposed to any of the drugs already had a mutation or a alteration in penicillin binding protein 3. I'll talk a little bit about what that is, but and that's when the Acinetobacter MIC was already a little higher. After treatment, then that same isolate essentially evolved to acquire secondary mutations. So now it already has that PP3 mutation, but on top of that, it acquired mutations that allowed it to increase drug efflux, and that probably contributed to the higher level resistance. After cefiderocol treatment, again, if you usually understand mechanism of resistance, you can probably connect the dots to understand mechanism. If you understand mechanism of action, you can understand mechanisms of resistance and vice versa. So after cefiderocol treatment, there was the initial 2 mutations, and then one of the more common mechanisms of resistance is cefiderocol, which is if cefiderocol is using an iron transporter to get in, then bacteria have a diversity of iron transporters, and they're able to turn some off. So there was iron transporter truncation, essentially, and that led to high-level resistance to both drugs. Now, you're noticing both drugs, even though they're a little bit different, what makes them almost identical is what happens once they actually get inside the cell. They're both, how they kill is by inhibiting cell wall synthesis, and they do that by targeting one of the main uh, proteins that is essential to building the cell wall, PVP3. But you can see how if there's a single target, how these drugs are a little bit more vulnerable to the emergence of resistance because you have to just change that one target. So our lab's overall goal is to try to understand on a molecular level and on an evolutionary level how these extensively drug-resistant organisms become resistant, and not just to Acinetobacter and cefiderocol, but in general to last-line agents, and then translate that molecular insight and knowledge to develop better diagnostic and therapeutic strategies. So today I'm actually going to focus on one cohort. So in order to achieve these goals, what we've done is tried to assemble a really large collection of MBL-CRE clinical isolates from around the world. So we have about 550 clinical isolates now, contemporary isolates from the last 4 years. These are different patient populations, but tertiary hospitals across 6 continents. Partly what makes it so hard to study MBL, like these type of bacteria, is that they're very rare still. They might be very common in certain settings, but now if you think about you're trying to find ones that are resistant, that's even more rare, right? So you need to get a pretty big collection to have statistical power. So today I'll focus on one cohort within that larger collection. So this is a single center cohort. So this is from MD Anderson Cancer Center in Houston, Texas, which is a higher level specialized tertiary care facility for cancer patients. And we'll just focus on the Acinetobacter resistance aspect. This was recently published, so most of the initial data is now available on AAC. So this particular cohort specifically includes 48 unique patient E. coli isolates. So these are NDM-producing E. coli isolates that were collected within contemporary isolates recently collected. And our overall approach was to try to connect the dots between what's happening on a molecular genomic level to what we're actually visibly seeing. So your phenotypic AST methods, whether that's broth microdilution, disk diffusion, and even other phenotypic assays that give us a little bit more information about exactly how antibiotics kill, right? Like MICs are a 24-hour endpoint assay. You see if something is growing or not in 24 hours or so, right, overnight. But then there's something called time kill assays, which allow you to understand more complicated kinetics, like how fast does killing happen and how much killing happens early. Microscopy gives you another type of information. And then connecting that to ultimately both combination of short and long read sequencing. The goal being connecting the dots between different types of data sets. So one of the first things this is just a single center cohort. I'm trying to keep it simple because I think sometimes even smaller cohorts are helpful. Just at this one hospital, MD Anderson Cancer Center, they observed essentially that there was a gradual increase in the amount of NDM producing E. coli that were isolated from year to year. And this is happening across the United States, so it makes sense. And don't get too lost in the figure. It's a complicated figure, but I'll guide you through what you need to focus on. So the first, one of the first assessments, these are 48 isolates from unique patients. One of the first assessments that the whole genome sequencing aspect gave us that it's not possible for us to understand with conventional phenotypic testing is that we have a better idea of epidemiology. So we saw that most of the NDM producing E. coli were high risk E. coli lineages. So these are known to be having a higher propensity to rapidly disseminate globally. They're more virulent, they're more problematic in different ways. And that's not a good thing. And then we tried to look for specific mechanisms of resistance. And the most common mechanisms of resistance that we're interested in our context is looking to see if what we've seen in patients, right, like the case that I just presented, PBP3 mutations that previously have been linked to clinical treatment failures, how prevalent are they in this one cohort? And we found that they were pretty common. And that was, at least for us, unexpected from a regional epidemiology perspective. So in this particular cohort, 88% of NDM5 producers had this mutation that has been linked frequently to treatment failures. It's really just that last bar you need to look at where there's a black box. Let me see if my laser moves. So wherever there's red, there's a PP3 mutation, right? So pretty much all the isolates, which to me was a higher prevalence than I expected. However, from our global cohort, we know that there's quite a bit of variability by region. The average is, if you combine all those isolates, the prevalence of PP3 mutations is 60%, which is still pretty high, but it varies drastically, anywhere between 10 to 90%, depending on what region of the world you're looking at. And I think there already has been quite a bit of work done from India itself to show that there's a high prevalence in some regions in India. And that goes to, almost the exactly same prevalence in MD Anderson Cancer Center. So it transcends continental boundaries. And with some structural modeling, we're able to see that these specific mutations that are related to treatment failures, what we think is happening is that these mutations or insertions are located not exactly at the binding site of where the antibiotic binds, but at the entrance. Essentially, the model allows us to predict that what's probably happening with this insertion is that it makes it a little bit harder for Asterium or Cifotericol to get access to where they need to go. One thing that I was really interested in, because we're trying to connect the dots between phenotype and genotype, is because PBP3 insertions are linked to resistance. We were surprised to find that majority of the isolates, so 61% of the isolates that had PBP3 insertions were categorized as Astyranamycobacterium susceptible. So this is when you get to, complicated territory, or what does that mean? What do you trust? What is real? What's the truth? So one aspect that is very helpful, even if you have a smaller data set, right? This is a single center population, but we did have clinical data available from patients. And that is very rare with certain organisms like this. But I think one thing that's missing is connecting the dots between in vitro data, animal models, and clinical data. Which is what we then did. And I can kind of guide you through that again. Keep in mind, even though these were 48 unique patients, about half of them are isolates that came from non-sterile sites, right? So it's hard to tell in a smaller patient population like this if a patient does well or they don't do well, whether that's really a result of the antibiotic treatment or because they're, the many other variables, right? It's hard to control for that. But one thing that was very statistically significant in terms of having a strong relationship is Astyranamycobacterium resistance was not linked to any single variable, but a combination of different mechanisms working together. We already knew PP3 mutations insertions are important, but it was a combination of PP3 insertions and these CMY beta lactamases that came together to essentially lead to high level resistance. The boxes are a little off, but I can navigate with my pointer. Hold on. Can you hear me still? I'll stop sharing and reshare. It was stuck, I think. There we go. There, that looks better. So all you need to see is there's all the purple is where there's PBP3 insertions, but that alone is not enough to correlate with Astyranamycobacterium resistance, which is here in the bottom. So that's a gradient from susceptible to more resistant to the right. So the combination of both PP3 insertions together and this other beta-lactamase called CMY is what was a better predictor of increased Acinetobacter resistance. And we looked at certain other variables. This is just showing specifically that even though, so in general, when we're worried about NDMs, we forget that there's a lot of other beta-lactamases that are also being carried by these organisms, right? Because NDMs are so bad. But these enzymes that are, we thought historically would be able to be taken care of by a lot of beta-lactamase inhibitors are now making a resurgence. CMY42 was a strong driver of high-level Acinetobacter resistance. So, and that was independent of NDM. So with NDM and no CMY on the left, and then NDM plus CMY, and that was a statistically significant association. And what was helpful looking at clinical outcomes is there were 7 patients in the cohort that did have repeat isolates. So that means that potentially that's a signal for clinical failure. So 7 patients, and all of them had invasive recurrent NDM coli infections. But across all those 7 patients, what was interesting was, and these are, again, the second isolate was obtained after they were treated with Acinetobacter, or in this case, ceftazidime-avibactam plus Acinetobactam. 6 out of those 7 patients, that second isolate didn't have any phenotypic change compared to the baseline isolate. In 2 patients, and I'm showing with the color, orange basically corresponds to patient 2 up here. Patient 2 did die within 25 days of the second isolate growing, and then patient 5 down here died within 6 days of their recurrent bacteremia. And I think patient 2 is more interesting because that was the only isolate that we saw a significant phenotypic shift in MIC, where the initial isolate was 16, and the second isolate that was cultured after treatment failure was greater than 256. And I think it essentially showed us that all of these case reports have these threads in common. The baseline isolate already had a PP3 mutation, but after treatment, there were new mutations, secondary mutations that were acquired in PP3, in efflux pumps, and other mechanisms that basically work together to lead to resistance. I'm gonna skip this part and get to the model. So what we think is happening, just based on the case reports that are coming out, based on our data, is that maybe phenotype is not enough to capture everything that we need to capture. So if you have a normal NDM E. coli that doesn't have PPP3 mutations, maybe you can treat it with Acinetobactam and the phenotype is reliable. But if you have a NDM E. coli that does have the baseline PP3 mutation, because in all of the cases of the treatment failures, the first initial isolate had the PP3 mutation. What we're calling it essentially is an adapted strain that already has something that leads it to be more evolved. And we call it the 2-hit hypothesis, which is inspired by, there's precedence for this in cancer biology, where 1 mutation is not enough to lead to the final cancer presentation. You need 2 hits, a combination of 2 mutation leads to cancer. So based on that similar paradigm, it's possible that whenever there's this baseline PP3 mutation, most of these isolates test susceptible. Their MICs are pretty low, so they may not show those phenotypic changes, but they may already be very primed and predisposed to evolve high-level resistance if the patients are actually treated with Acinetobactam or cefiderocol. So if the patients are treated with that, what we're seeing from the case reports at least is there's pretty rapid selection of high-level resistance. And that's due to the acquisition of now second hits or secondary mechanisms that together lead to high-level resistance. So I think this is essentially showing us that this evolution is happening within a patient. And last 2 slides, I talked about diagnostics, but an example of how translating mechanisms of resistance can be helpful for therapy is the example of using combinations of antibiotics based on your knowledge of resistance. So if we know that PBP3, the single target being mutated, is problematic and that is a driving force of resistance, then the natural hypothesis is, what if you target multiple PBPs? So there's a drug right now that's designed for a different organism, Acinetobacter. It's part of sulbactam durlobactam. But the way it works is it is a beta-lactamase inhibitor that also binds PBP. So in addition to our hypothesis essentially is that what if we combine Astrionum, which is currently available, with durlobactam. Astrionum binds PBP3, as we talked about, and durlobactam is able to bind PBP2, which is a different PBP. This is the last thing that I'll end on. Our prelim data essentially does show that for isolates that are fully resistant to Astrionum avibactam and cefiderocol due to PBP3 mutations, the approach of inhibiting multiple PBPs does work in overcoming that resistance. And we wouldn't have been able to take that approach if we didn't understand mechanisms of resistance. So Astrionum durlobactam might be a viable treatment option. So in conclusion, I think in general, susceptibility testing, even what we have right now, is very important in order to guide treatment decisions. A lot of the treatment failures happened in patients where the baseline isolate already had elevated MICs. But we also know that there's different aspects of resistance that might not be detected with just MIC testing. And if we understand that, then maybe we can actually build better diagnostics, better treatments. And also in this case, I think it's being more precise about when we want to use our existing antibiotics that are already available to us so we can extend their life expectancy. And with that, I'm going to stop and I think we can get into the discussion. Thank you so much.
Thank you so much, Dr. Ayesha. Very insightful and very well represented with real life case scenarios that made it all the more interesting. Thank you so much, Doctor. It was very insightful, very helpful. And we now know we have someone we can fall back upon whenever faced with queries around antibacterial resistance, and we will know we have the expert opinion. So, analytics webinar session. I'm sure the audience enjoyed it to the utmost. It was very, very informative. Have a great day and see you soon.